Hey guys, today we're going to start 2.5 enzymes. You know something is an enzyme if it ends in ASE, and we'll talk more about that in a second. But the reason we talk about enzymes right after we talk about proteins is because enzymes are proteins. And this is an interesting topic because you're going to do the first practical uh, in your SL Bio class. So there are 7 practicals that you do over the 2 years. They're kind of like labs. And you're going to do this one virtually, which makes it a lot easier. And so let's get started. So, understanding number 1, enzymes are proteins that have an active site to which specific substrates bind. Okay, so enzymes, right? They're proteins, and their job is to catalyze reactions. That is, to speed up specific chemical reactions, whether they're anabolic or catabolic, by lowering activation energy of the reaction. Okay. Each enzyme has an active site that allows it to bind to a specific substrate. So if this is your enzyme, you'll see this shape here. This is your active site, and then this is the substrate. This is the thing that the enzyme works on. It either splits it apart or it binds it to something else. And you'll notice that it's got that shape that fits in here. So when I say in biology, your form is your function or your shape is your job, this is what we're talking about. You have an enzyme that's got a very specific shape. It only fits this specific substrate, right? And then when it's bonded together, you call that the enzyme substrate complex. Okay, so here's your enzyme, here's your active site, here's your substrate. Now your enzyme has bonded to your substrate. We call that an enzyme substrate complex. But let's back up for a minute because I said a minute ago enzymes are proteins that speed up specific chemical reactions, meaning whatever reaction is going to happen to the substrate was going to happen anyways, but this enzyme makes it happen faster by lowering the activation energy. So you all took chemistry last year, right? But I don't remember how much you know, or I don't know how much you know. So let's review what activation energy is again. So, activation energy is the minimum amount of energy required to initiate a chemical reaction. So, here we have both endothermic and exothermic reactions, right? And that's whether you take in energy during the reaction or you give off energy during the reaction or after. So, it doesn't really matter whether you're talking about endothermic, which means energy in, or exothermic, meaning energy out, right? What we're talking about is activation energy, and that's this part right here, okay? So, your activation energy is the amount of energy it takes. Here's energy on the y axis. It takes to get this reaction really going. This is the direction of the reaction, right? So, you're producing products that way, producing products that way, okay? So your activation energy is the amount of energy required to initiate that chemical reaction. It's like you got to roll the rock up the hill, and then the reaction is rolling down the hill, right? So how high up the hill do you have to push it? Now, if you have a catalyst, that's going to reduce your activation energy. So you don't have to spend as much energy to get your reaction going. That's why enzymes are so important in the human body because, like I said, you are basically a walking sack of biochemical reactions. And enzymes, they're there to save you energy, right? They save you all this extra energy it would take to get that reaction started. Okay, so enzymes, they reduce the activation energy to get a chemical reaction started. Okay. Okay. Understanding number 2. Enzyme catalysis involves molecular motion and the collision of substrates with the active site. Well, we all know that molecules vibrate, right? So when we talk about temperature, we're talking about how much a molecule vibrates. So in order for chemical reactions to occur, the reactants, the two different atoms or molecules have to bump into each other, and they bump into each other because they're vibrating all over the place. So, the structure and chemical properties of an enzyme's active site is complementary to the substrate molecule it binds to. Okay, that's a fancy way of saying that the substrate fits the active site of the enzyme, right? Because here's our enzyme. And here's the active site, right? And here's our substrate. And check it out, our substrate fits our active site, okay? In order to catalyze this reaction, first the substrate has to collide with the enzyme's active site, forming the enzyme substrate complex. So we know that. Here it is. Here's the complex, right? Now, something we should probably talk about that you may not be aware of is the enzyme actually bends a little bit to accommodate the substrate. So it's a good fit, but it's not exactly perfect. So the enzyme does bend a little bit to fit the substrate, right? And you can kind of see the change here. Next, and this is if we're talking about an anabolic, or nope, this is catabolic, right? So we're talking about a catabolic reaction right now. The substrate's broken down by the enzyme. So here's our first step, second step, here's our complex, right? And then the products are released into the environment. So let's see. And then the enzyme is unchanged. All right, so here you go. It breaks these two things apart, right? Now your products go out into the environment. So this is step 3, right? And then your enzyme, it's back to the right shape and it can start all over again. So your enzymes can be reused over and over and over again. Like you don't throw out a pair of scissors after you use it one time, right? You keep them for the next time that you need them. And enzymes work the same way. So enzymes go through this process repeatedly. So the same molecule is used over and over and over again. Okay. So one more time, you have substrate and you have your enzyme. Step 1, right? They come together. The enzyme changes shape a little bit. The enzyme then breaks them apart. The products go out into the environment, and then the whole process starts over again. So, a good catabolic example is when you eat food, right? So, when you're eating food, you get thirsty, you have hydrolysis. Now, we said before hydrolysis is just the addition of water to split up polymers, right? But it's not that simple. It's not just water. There's an enzyme that we add in there too that helps speed the process along. And when we talk about digestion, you'll learn about all the different types of enzymes that do that. So, amylase breaks down amylose. So, this is amylose. You know it's a carb because it's OSE, right? Here's amylase. You know it's an enzyme because it's ASE. And your amylase works like scissors and breaks those apart. And you should also get a drop of water coming out, right? Because it's hydrolysis. Oh, wait. So water doesn't come out, water goes in. My bad. You have to add water plus the enzyme to get it to break. All right, so amylase plus water helps break this down. Okay. An anabolic reaction or building something up, because enzymes also build, right? They're the doozers. I don't know if you guys ever watched Fraggle Rock, but enzymes are the doozers of your body. They build stuff up and they break stuff down. So the anabolic example is here we have, 2 amino acids. How do I know they're amino acids? Well, here's the butterfly. Let's review the 3 parts, right? We have the amino group. We have, it looks like just a plain old H for an R group. We have our carboxyl, right? And we see the butterfly. So we know that that's an amino acid. So here's 2 amino acids that we're going to bond together to form a dipeptide, right? And we know that we always connect our what? We connect our carboxyl group to the amine group to form that dipeptide bond, right? Or the peptide bond. So let's see, did I do it right? I didn't do it right. Look, I did it wrong. That's not good. Let's fix it. No. That's no good. No good at all, I say. Uh, hmm. I don't really feel like messing with this right now. Anyways, let's show it again. All right, so we know that it should actually be this one right here, right? Carboxyl group to the amino group. So, my bad. All that's really happening is that this line right here, this should be pointing to that. That's all. All right. So when we talk about enzymes, right, you often hear what's called the lock and key model, and that means that only one specific substrate can bind to the active site at a time. They fit like a lock and a key. So if your substrate, the stuff that you're building up or breaking down, is the substrate, that's the key, right, and your enzyme is the lock. And you can think of it as like the lock is the active site, right, and your substrate is the key. And when the key is in the lock, that's the enzyme substrate complex. Another example, we're going to use amylase again. It's an enzyme because you have ASE, right? It breaks starch, particularly amylose, into simple sugars. And it only works with specific sugars. So here's glucose and glucose, right? We're going to use our amylase, and it's going to break those two apart, right? But now if we try and use our amylase on fructose and glucose, it doesn't work, right? It only can break the bond between two glucose molecules. So we cannot use it on other molecules. But we can reuse that amylase. So we're going to break those apart. And we're going to break those apart. Same scissors. You don't throw your scissors out. Okay. Now, enzymes aren't always effective. Remember, we talked about how proteins can change their shape. Well, you can probably guess that if enzymes change their shape, they're not going to be able to do their job. So, the function of an enzyme is to increase the rate of reactions, but their ability to do so can be affected by a couple of different factors in the environment, particularly temperature, pH, which is, acidity or basicness, and substrate concentration. Now, when we say substrate concentration, this is how much of the substrate there is, okay? And that's exactly what your lab is going to be all about. So, enzymes catalyze reactions at an optimum temperature, and that's typically related to the environment. Remember, enzymes are proteins, so high temperatures can cause the enzyme to denature, and when they denature, they can't do their job anymore. They don't fit. That active site is the wrong shape. So, if temperatures become too high, right here, right, you've got denaturing happening. The enzyme is changing shape and it's no longer functioning. So the enzyme activity slows down. It's not as effective. But as your temperatures fall, right? Or if you have cooler temperatures, that's all here, your enzyme activity isn't going to be at its peak because remember, chemical reactions require the vibration of those molecules because they have to bump into one another. So as they slow down, they don't bump into each other as much. So you don't get your optimum rate of reaction. And this is exactly how your body uses a fever to ward off infection. So when your body temperature is normal, right? 98.6, your enzymes work just fine, doing their thing, breaking stuff down. But if you have a fever, if your fever is too high to the point where it's damaging not only the invader's proteins and enzymes, but it's invading or it's denaturing your enzymes too, there's a huge problem. You're not going to be able to break things down the way you need to, and you're not going to be able to speed things up. Now, this can be a real problem if you don't have the ability to break down compounds to generate energy. So, if you can't undergo cellular respiration, that's a big deal. Your cells can't do all those processes that they need to do to keep you alive, right? So, that's why fevers are good. Fevers are a sign that your body is fighting infection. But too high of a fever can actually damage your proteins and your ability to fight infection. And usually we consider it dangerous if it's anything over 103, I believe. 2 is not great. 2 is bad. 3 is like super bad. Now, that doesn't mean that all enzymes have the same optimal temperature. So, remember we talked about how different individuals and different species have different proteomes, or they have the different range of proteins that they can produce. Well, enzymes are proteins, so we all produce slightly different enzymes, and those enzymes have different optimal ranges, right? So here we have three different types of bacteria, and we have sugar, and we have temperature. So we have how much sugars are being broken down by these bacteria. So here you can say for this particular bacteria, its optimal temperature is about 60 degrees. For this, for T fusca, it's going to be here. And then for C, thermocellum, thermo meaning hot, it's quite a bit higher. So that's a big range. That's like 20 degrees difference in terms of optimum temperature. That's a big deal. It's a good indication that this guy down here, he's probably adapted to moderate conditions, right? So he's still got a pretty high range of functionality, but he probably thrives the best at about 60 degrees, lower than room temperature, right? Room temperature is about 70 degrees. So he lives in moderate to moderately cool environments. T. fusca, this is the stuff you find in the house, right? So this guy is his perfect temperature for breaking down sugars is 70 degrees. He's adapted to nice room temperature environments. Now, C. thermocellum, this guy is not happy unless it's right about 80 degrees. So that's a good indication that this guy, thermo, is adapted to warmer conditions. Now, you'll also notice there's a pretty steep drop off right here. So this guy's living pretty close to the edge, right? Too much higher than that, and he's not going to be able to break down those sugars. So you can often get a good indication as to where different species live based on their optimal temperatures of their enzymes. Or you could work it the other way around. You could make predictions about what the optimal temperature ranges are for their enzymes based on the environments that they live in, because this is natural selection working, right? So you are better at surviving in conditions that you are adapted to. If your enzymes are uh, adapted to working in a particular set of conditions, that's where you're going to live, right? Enzymes also have an optimal pH, meaning they work the best at a specific pH. Now, when you deviate from that, and we kind of foreshadowed this in our last lecture, I gave you a question about what happens to a particular enzyme that's meant to work in the stomach. What happens if it ends up in the small intestine? Because the pH conditions are very, very different. And for the most part, you all got it right. It's going to denature. It's not going to be able to function in the intestine because it's designed to work in acidic conditions instead of basic. So, for example, protease, I think that's the one that we used. Protease is actually, no, we didn't use protease. We used something else. Anyways, protease is very, very basic. right? So it likes to function way, its peak is like that. So it functions over here. So that would be a stomach enzyme. Whereas other enzymes tend to be, wait a second, something's wrong here. I must have been like not paying attention when I made this PowerPoint. Let me fix this. 1.6 is not basic. There we go. Embarrassing myself. Okay. So stomach protease, I knew that wasn't right. Stomach protease is acidic, right? So stomach, it's going to be, I know my enzymes, acidic protease, right? Whereas other enzymes that function in the small intestine are like this, right? So their optimum is like way over here. So if your enzyme is not working in the proper temperature or in the proper pH conditions, it's not going to function and it's not going to do its job. Just like if you're not happy, if you're tired or you're hungry, you're not going to get your work done because you're tired or you're hungry, right? The conditions have to be right. And that becomes a big deal when you're talking about multicellular life. The whole reason you see the diverse types of life and forms of life on our planet is because organs are basically segmented areas that provide certain conditions, certain pH conditions, certain temperature conditions, certain osmotic conditions that allow particular chemical reactions to occur where they are. So your stomach is very acidic, but your intestines are very basic. Those two different conditions allow different reactions to happen. You rely on both of those conditions, right? For those particular chemical reactions. So your body needed two separated places for those reactions to happen. And that's why you have all the different parts and pieces that you have to accommodate all these different conditions that are required for chemical reactions. So we're going to take a look and see exactly how this works visually because I'm a visual person. And this PowerPoint is available for you. So you can click on that link yourself too and play with this. Yep, Lab Exchange. Also, while we're doing this, you guys should sign up for Lab Exchange. It's a really great source for information. We'll be using it for labs. So I'm going to walk you through that real quick. Okay. So the website is labexchange.org. So if you just type in labexchange.org, you'll come to the Lab Exchange website. Now, mine, it automatically takes me in because I'm already logged in, right? And then you're going to choose that you are a student and you are going to sign up for my class. Am I signed in? Yeah, I am signed in. Dashboard, classes. Okay, here you guys are. So, junior bio, here you go. Your class code is 852500. You will register for the class. It won't let you in immediately. I have to allow you in. So, one thing I would like you to do right now is I would like you to go to Lab Exchange. I would like you to sign up as a student and I would like you to register for my class. The class code for both sections of junior bio, so that's both 1.3 and 1.4. Your class code is 852500. So when I look at this, the next time I log in, I should see like a whole bunch of people that I need to approve. Okay, so make sure you do that. All right, so where was that? Let me click on that link again. Okay. So here we have an enzyme model. We have a catalyst and we have molecules. So here's our two molecular compounds. We have A, which is this green thing, A2. Here's 1A. Here's another. They're bonded, A2. Here's B2, right? B2. Now, there's a chemical reaction that's going to happen, and we're going to form 2AB. So I'm probably bringing you back to chemistry right now. So we're going to run the model and observe what happens without a catalyst. Then we're going to pause it. We're going to add 3 or 4 catalysts in there by clicking the button. And then we're going to run the model again and see how the catalyst affects the reaction. Okay. All right. So we're going to play. No catalyst right now. And here our molecules are vibrating, bumping into each other, doing their thing. And this is taking a while. Nothing's happening. Nothing's happening. We're looking at picoseconds here. All right. I don't feel like waiting that long. So let's see. What happens if I increase this? Can I increase this? No, I can't. Still nothing. Oh, wait. There's one. It's broken. There's, okay. Oh, and they're back. Dang it. Why would you ever watch this in slow motion? It's taking for. Oh, there's one. It's broken. Still no AB though. So we have an A that's broken apart, an A2 molecule that's broken apart, but no B2's broken apart. We're just going to watch till we get our very first AB. Because dang, this is taking forever. Still nothing. And that A2 went back to being an A2. Okay, this is taking a stupid long time. Let's add some catalysts and speed this up, shall we? There's one catalyst. Okay, so our catalyst, it formed a bond. The active site formed a bond with an B2, and it's going to break that B2 apart. There it is. So there's the other one. So now we have one B2 free, or one B free. Still taking a while. Let's add another one. Oh, let's see. It said add 3 or 4, so let's add 4. Now we're cooking. Look, we're actually getting products now. Look, you can see our things climbing up. So we couldn't just add one enzyme, right? Wow, look at it go. So now that we've added these little doozers in here whose job is to break up and reform things, we added that catalyst. Looks like these guys are just, it's catalysis, so they're just breaking them apart. They're not adding them together. They're combining on their own. But we needed these catalysts to get that reaction started. It reduced the amount of energy, the reaction energy, to get the reaction started. Yeah, much better. We're not quite to completion yet, but you get the idea. All right, so if you feel like playing around with the simulation, if you once you sign up for Lab Exchange, if you just search for enzymes or catalyst, this simulation will pop up and you can play with it, or you can just click the link in the PowerPoint. Okay. So, substrate concentration, that's the last thing that we're going to talk about that affects the completion of the reaction, right, or reaction rate. And that refers to the amount of substrate that's available for the enzyme to work on. So, if there's not a lot, so say you have an enzyme and its job is to break up a carbohydrate. If there's not a lot of carbohydrate there to break up, right, it's not going to have a job to do. So, as the concentration of the substrate decreases, the reaction rate decreases. That makes sense. If you're a cookie monster and you're eating cookies, right? If there's no more cookies to eat, you're not going to eat more cookies. Yeah, that makes sense. Okay. However, let's take it the other way, right? So let's say you've got a whole lot of substrate, you have a whole lot of the building blocks or the materials, right? For whatever the reaction is going to be. Now, the enzymes can only work as fast as they can. When all of the enzymes are saturated, meaning they are bound to substrates, they physically can't do anymore. It's like having only so many workers and so much work, right? If the work is infinite and you only have so much workers, that rate of the reaction, it's going to hit a peak and then it's going to stay there because those enzymes can only work as fast as they possibly can. Right? They can't work any faster, so the reaction rate stays the same once they're all saturated. And we call that point where it starts to level off, we call that the point of saturation, meaning all the enzymes are busy. That reaction rate can only increase so much. So here we have some enzymes are busy, more enzymes are busy, more enzymes are busy, more enzymes are busy. Eventually, all the enzymes are going to be busy and they can't work any faster. So that rate of reaction is going to stay the same. Just like we talked about last time, enzymes can be denatured when they're exposed to conditions that interfere with their primary sequence or their primary structure, or their secondary structure, or their tertiary structure, or their quaternary structure. They probably don't mess with their primary structure that much, actually. It looks like mostly secondary and tertiary, right? So it's messing with its two-dimensional and three-dimensional structure, not the order of the amino acids. So each enzyme has optimum conditions in which it most effectively catalyzes reactions, and those are usually related to a habit of an organism's cell or a location within the body. And that's kind of what I was talking about earlier. So you have enzymes that belong in certain organs because they do certain things and they need certain conditions to do those things. And if they don't, they're going to get denatured and then they don't work. So look at that. We're all the way up to understanding number 5. Enzymes can be free or immobilized. Immobilized enzymes are used in industry. So we use enzymes not just in living things, but we use enzymes like everywhere for a ton of different things. Immobilized enzymes are those that are attached to a specific material so that it stays in place. And when you immobilize an enzyme, all it does is it means you don't lose it, right? So it's usually bound to a solid or a porous material, or it's mixed in with like a matrix, or it can be embedded in a soluble membrane. And these enzymes aren't changed in the reactions, so you can use them over and over and over again. We use immobilized enzymes to capture carbon. So as polluted air or air with lots of extra carbon in it passes over the enzymes actually grab and trap the CO2 for storage. And we're using that to create recaptured carbon. It's like solid carbon to reduce emissions from factories. You see them on smokestack scrubbers too. Now, if you are lactose intolerant and you drink lactose free milk, that's because of immobilized enzymes. So what happens is you take lactase, right? Lactase, ACE means enzyme, and you know it breaks down lactose, which is a carbohydrate, right? Immobilized enzymes are also used in producing lactose-free milk. Lactase is immobilized in lignite beads, so it's kind of like a polymer bead, like a gel bead, sort of. And then think of like tapioca, like when you get bubble tea, it's kind of like that. And in that bubble, there's a bunch of enzymes stuck to it, right? So then when you pour regular old milk over the beads, the lactose binds to the enzyme. So if here we have our enzyme, right? Here's our lactase. And as you pour the milk, the lactose molecule, that's not really what it looks like, but binds and it breaks that lactase apart into pieces. And that's how you get lactose free milk. Now, it's important that these beads are immobilized because you don't want to like have to constantly make more beads so you can reuse these beads over and over and over again. So we use enzymes in lots of industrial practices. And if you ever eat food that has a particular compound or protein removed or carbohydrate removed from it, it's usually done via enzymes. Or like milk and cheese, we use enzymes to produce milk and cheese, any kind of like dairy stuff. Enzymes are where it's at. One of the reasons why I think I told you guys this. So I'm celiac. I can't eat wheat. And it's not that it's not an allergy. Like I'm not allergic to it. The problem is my body can't physically break down the gluten. Gluten is a protein. So if you know someone that's celiac, gluten is a protein. Um, and some celiacs have immuno responses, but most celiacs are celiac because they don't produce glutinase ACE, which breaks down gluten, right? So you get this big funky molecule with a million branches, and your body physically can't break it down. And when it gets absorbed into your intestines, it like shreds your intestines because it's too big. So if we had the enzyme that could break it down, then you could eat gluten. Unfortunately, they don't make enzyme pills that you can just take because it would be denatured in the pH conditions in your stomach. So, another example of enzymes being denatured. That's why you can't just take a glutenase pill and be able to eat bread because the acid conditions in your stomach would denature the protein or the enzyme. Unfortunately, it doesn't denature the protein. All right, so here you go. Because lactose-free milk has more sugar monomers, it tastes sweeter. So if you don't like milk, try drinking lactose-free milk. Manufacturers also use this to sweeten dairy products without adding extra sugar. So if you have like a particular kind of milk, like a lot of people like TG Lee milk because it tastes better. It tastes better because they're using a little bit of lactose to break down some of the sugars in there, and it tastes sweeter. So they're not adding sugar; they're just changing the chemical composition of the sugar so that it tastes better. Okay, here are some review questions. Most of these are embedded in the lecture themselves. Also, be aware that you should be able to draw these graphs, right? So temperature, it's probably going to look like this. pH, it's going to look like this, or it could be like this. But substrate concentration, it doesn't have that peak, right? It's got a plateau, and that's the saturation point. So remember that. You will have to draw those graphs and understand what they mean. And in your lab, actually, you're going to spend some time with graphs. So make sure you're doing all this stuff in your notebook along with meeting the objectives for 2.5. And now let's get started. I'm going to walk you through the lab that you're going to do. Okay. Nope, I don't want to keep that. All right, so let's go to Canvas. Let's pick on 1.3. Okay, so you're going to click on, just to show you where we were in topic 2. We were in enzymes. You're finishing up the Edpuzzle lecture right now. Oh crap. Where's your, oh, I know why. So here's the 2.5 understandings and skills, right? So just remember all this stuff is what you need in your notes. Make sure you can answer all these questions in your notes. Here's the PowerPoint that we just went through. Now you're going to go back to modules, or you can even go back to the home page. And you're going to click practicals. Give it a minute. Okay, so here, these are the 7 major practicals that we're going to do between this year and next year. This is our first one. We start at practical 3. Don't ask me why, we just do. And you're going to click on this. Okay. So this is an assignment. The lab is designed to answer the following questions. How do substrate concentration and pH affect enzyme-controlled reactions? So we just talked about it in the lecture to give you some background, but now you're actually going to do it. So to get started, you're going to click on this link, and it's going to take you to a simulator. Make sure you have Flash enabled. Yes. Okay. Right. Here's your simulator. It's going to tell you to read the instructions over here. Right. Objectives, blah, blah, blah. Procedure. Okay. Before you start there, it's going to tell you follow the procedure in the right-hand column. Watch the brief video by clicking on the information button, or right, the brief video and clicking on the information button. Okay. So you're going to read this procedure, click the TV.
Enzymes enable molecules called substrates to undergo a chemical change.
Yeah, so you're going to watch that whole thing. And then you're going to click information, and it's going to talk about some stuff, clarify some stuff, maybe that I didn't talk about so great. Okay. Now, on a sheet of paper, you're going to create a table that looks like this. Or if you're on a computer, you can create your own table that looks like this. The reason why is because when you go through the lab, you're going to have to reset it. And every time you reset it, you're going to lose your data. So you're going to write down and collect your data as you go. So procedure, set your pH values on your test tubes 3, 5, 7, 9, and 11. Keep your temperature set. Okay. pH, test tubes. Okay, that's annoying. There we go. 3, 5, 7, 9, 11. Okay. Put 0.5 grams of substrate in each test tube. 1, 2, 3. Oh, it's changing different colors. 4, 5. Okay. Now what do I do? Click on the computer monitor to show your results. Okay, so here we go. So here's our test tube number, right? That's the amount of substrate that we put in. And then this is the number of molecules of product formed per minute. So this is the reaction rate. Ready? How much? Or basically, it's you got 19 molecules of product per minute, 39 per minute, 72 per minute, 45 per minute, 24 per minute. Now you're going to take this information and you're going to write it in your table. You would write it across here, across the top. Now, there's also a table down here, but if you like type it in here and then go back to do the next part and hit reset, you're going to lose it. So don't put it in here until the end. Okay. So you're going to repeat that process. Oh, here, let's click the reset button. Reset. Okay. You're going to complete that process and record your data for all the different substrate concentrations at the different pHs. Okay. Now, in real life, you would have to do actually a lot more than that. You would have to do uh, each one 5 times and average the results. But because there's no variability built into this simulator, we don't have to do that. So you can just fill out the table and do it one time. Once you have this complete table filled out, right, then you open this up and you enter all your values in here. And then you click print. You can save it as a PDF, right? It'll be a nice, pretty table. And you're going to save it to your desktop because you're going to upload it to this assignment to get credit. Okay. So print that out, save it. You don't have to like print it out on paper, just print to PDF so that it saves on your computer. All right. And then you're going to hit graph and it's going to produce a really nice graph for you of all of your results. Okay. And you're going to do the same thing. You're going to hit print and you're going to save it as a PDF. Mine doesn't show up because I didn't do the whole thing. All right. You're going to save it as a PDF and you're going to save it on your desktop. Okay. And then you should be able to, there'll be a submit button here and you can submit those PDFs. And that's how you get your 10 points for this part of the lab. Okay. Now, make sure you hold on to those PDFs. Don't get rid of them yet because hit next. And the next thing you're going to do is a quiz. And it's like 5 questions, super short. Don't panic. You're going to use your lab table and your graph that you just made and your notes if you need it. And it's just 5 questions. And by the way, make sure you close all your other tabs before you start because remember, we can see when you're like clicking off and look at stuff. So just close your other tabs and then you won't have to like explain to me why you were clicking around on stuff. You shouldn't need anything except for the PDFs. I'll see when you click off and look at those, right? But just close all your other tabs and then you can take the quiz. And it's 5 questions. Make sure you're answering thoroughly and completely because this is for correctness. It's not just like, type some letters in the box. And all this is due, I believe, on Friday. Okay? Check the deadline. It might not be Friday. It might be before that because Friday we're doing a case study. Woo! We're going to do an actual vet case study with enzymes and it's fun. Alrighty, so that is how you do both the lab and the practical itself. You're going to learn all sorts of fun stuff about enzymes and how they work. And remember, if you need to talk about stuff, if you need some help understanding stuff, by all means, send me a message, get a hold of me on Zoom, get a hold of me on Remind. We can talk about whatever it is that you need. Oh, yeah, due October 2nd, it looks like, which is Friday. Yep, okay. All right, and I think I will be live with you guys on Thursday when we actually do our case study. Okay? All right, well, have a wonderful day, and I will see you live and in person on Thursday. Bye, guys.